Ram, Jai Sri Ram, Jai Sri Ram. Culture media is artificial food, is used for growth of the microorganism like bacteria. Now this culture media we can divide, on in two types. First is minimal media, another one is supplementary media. There is a great difference between minimal media and supplementary media. Actually, the minimal media is that media which contain basic things. Minimum growth material is present, like they have some source for the carbon, like sugars are present, like glucose is present, okay, water is present, uh, protein is present, fine. But in this media, wild kind of microorganisms will grow, and that wild kind of microorganisms are called prototrophs. Fine, they are called prototroph. Another type of media is called supplementary media. In this supplementary media, we will use one selective agent, a specific agent we will use, right? And the microorganism will grow exclusively in the presence of that particular supplement, right? So now question is that as compared to wild type, this another type of microorganism, why it's need a selective kind of, is why it's need additional kind of things, right or not? So apart from minimal media, all kind of basic things present there, source of carbon, source of nitrogen and water and all. Apart from that, they need one additional thing. Why they need? Actually, the microorganism which grow here, they are called autotroph. What is difference between prototroph and auxotroph, students? Prototrophs are wild kind of the strain of the microorganism which grow in minimal media. They for synthesis of all kind of material, they have active gene and they will synthesize, fine or not? But auxotrophs are those microorganism whose some specific gene due to mutation, it's lost. As for example, I have one microorganism that is autotroph, that is wild strain. Okay, now its one gene got mutated and it's lost capacity to synthesize arginine, fine or not? So we will now, this prototroph will convert into auxotroph, and this microorganism cannot survive without that arginine added in that culture media. So supplementary media have minimal media plus arginine, then we will call it supplementary media, fine or not? So we have one prototroph, it's due to mutation it's lost capacity to synthesize specific amino acid, so they need of supplementary media, fine or not? As for example, I'm taking one auxotroph and I'm growing in minimal media, it won't grow. But when I will in that culture media and minimal media, I will add arginine, then this arginine auxotroph whose arginine synthesizing gene was mutated, it was inactive, they will grow and they will form colony. That means auxotroph, they need some specific, due to mutation they lost the gene and they need some specific supplement. Prototrophs are wild, they don't need any specific kind of supplement.